Screen, yes. Okay,
Okay.
So let's just wait a couple of minutes, and then we can start in 1-2 minutes. So it shouldn't be a problem.
Mm-hmm. I mentioned to Alexandra that I don't think I will be able to see any comments or in the chat room or something because I have only one screen. So if there is anything that to my attention, you please help.
I will. So I will let you know if there are some questions or anything that should clarify. Don't worry about that.
Okay, great.
Yeah.
Yeah, I can totally see why 2 screens would be better than 1, but anyway.
So we can start if you want.
Okay, great. So it's a great pleasure to be part of the Summer of Machine Learning at Skoltech. Today I will talk about machine learning in neuroscience, both classical and deep learning methods, and how they can help us understand the human brain and open a window to human cognition. Before proceeding, I would like to introduce the main non-invasive brain imaging technology that we use, and it will be the source of most of the data presented in this talk. Magnetoencephalography is a non-invasive brain imaging method that measures magnetic signals emitted from the human brain. The device that we have in the lab I oversee at MIT is shown here. I believe you can see my cursor. So it has a helmet structure containing about 300 sensors of magnetic fields. And these are very, very sensitive sensors. They can measure signals even 1 billion times weaker than the magnetic field of Earth. And this is why the device is inside a shielded room. During experiments, participants sit on this chair. Their head is surrounded by the helmet. And when there is brain activity, electrical current flows through the neurons of the brain. And the current produces magnetic signals, very, very tiny magnetic signals that are picked up by these sensors. And we can measure these signals dynamically and reconstruct activity on the brain, on the cortex, producing dynamic maps of brain activity. And these are the signals that I will often present today because that's my specialization as well. It's very clear to understand the power of this method by looking at some example data. So here we can see the pattern produced in the sensors in red and blue. Red is outgoing magnetic signals and blue incoming magnetic signals to the human brain. This is a flattened helmet surface. And this is in response to auditory stimulation to multiple times in an experiment. You can see how from this topography we can end up reconstructing brain activity on the cortex, showing here highlighted both in the right and left hemispheres, activation of the primary auditory cortex. This is the cortex that receives the signals before they're further processed in the brain. Importantly, you can also see time courses below of the response in this tiny brain area, seeing a deflection about 100 milliseconds from the onset of the stimulus. So this method is very specific in time and reasonably specific in space as well, offering us a very powerful way of seeing brain activity, measuring signals all around the brain. Another example, and I hope you can see the video. The bandwidth is good enough to see what is happening. You see here the time from the onset of the stimulus. The participant saw an image of a face, and you see here the dynamics of what is happening in the brain while the face is processed in the human brain, initially starting here, and then a cascade of activity further down the human brain as we're processing what we see in front of us. And this is exactly the kind of signals we often study with this method. And there is a lot of effort to study human vision. It's the best studied part of the human system. And nowadays, we are seeing an even more interest in studying human vision because of the great advances in machine vision. So let me start with some information about classical machine learning and how it is used. It really offers a lot in how we analyze the signals, and then I will proceed to also deep learning applications in the second half of the talk. So the main method of analysis of neuroimaging data is univariate methods, meaning that every variable is analyzed separately as an independent piece of data. So every sensor or every voxel is seen as a separate variable, and we study effects. And because you have so many of these variables, you often hear about mass univariate methods, meaning that you conduct univariate tests in a massive scale, one after another, constructing maps. However, a more powerful method of looking at the data is multivariate methods. And that's because information is contained not just in a single voxel or sensor, but in groups of variables. And this is why it is important to study these patterns. You may often hear this as multivariate pattern analysis or multivariate classification, multivariate decoding. These are not exactly the same, but for the context of this talk, it will be the same, though decoding is more specific than multivariate analysis, which can also have encoding techniques. So why do we need multivariate methods? So for example, let's say you have one source or one sensor in the brain, and we measure signals in response to condition A and condition B. There are cases where either the source or the sensor really doesn't give you any discriminative power between the two conditions. You don't see a difference. However, if you have a pair of sources, or more in the general case, and you see these data scatter plots in multidimensional space, you may be able to find hyperplanes or separating manifolds that really show the difference between the two conditions. So in this case, you will be able to discriminate condition A and condition B by using the combination of the two sources, but none of the marginal distributions of the individual sources would be helpful. And the same idea applies to sensors as well. So that's why the multivariate methods are so powerful, and this is why we use them now in MEG, magnetoencephalography, or MEG, more and more and more. These methods became popular the last 6 years or so, and they're increasingly becoming a tool of choice to see this kind of signals. So as I mentioned before, we have several sensors measuring magnetic signals, each of them producing a time course of magnetic signal. So you see several time courses here, one per sensor, and at every time instance, you get a profile of how brain activity looks from outside the head. The magnetic signals emitted from the brain as you're being recorded in the device. And what you can do is concatenate these signals in what we call a pattern vector. The order doesn't matter, but we get in about 300-dimensional vectors. And now it goes to the typical terminology of machine learning, meaning that we have several of these pattern vectors because when we conduct an experiment, we measure the responses several times. We present the stimuli many times because the signal-to-noise ratio is very weak in general. So in this case, we have several trials, as we call them, of presenting the same image. We call it condition A, and several trials of condition B. And these pattern vectors are coming exactly in the same idea. So let's say they're all coming from 100 milliseconds from the onset of the stimulus, marked as time 0 here. So all of them correspond to 100 milliseconds from the onset of the corresponding image stimuli. And what we do is separate these patterns as training and testing sets. I believe these ideas should be clear to the audience today because of machine learning expertise. And what we do is then we are training classifiers. In this case, and for reasons I will explain later, we're talking about a simple linear classifier such as a support vector machine and to get a decoding accuracy, and then apply cross-validation, meaning that we randomly reassign the data to training and testing sets and recompute again in a cross-validation way, eventually ending up with a classification accuracy showing us how well we can discriminate these two stimuli from brain signals. And one can repeat this procedure exactly the same way for every time instance, millisecond by millisecond, to extract what we call a decoding time course. So you repeat this procedure separately for every time point and get the corresponding decoding accuracy. And looking at this time course, you can really get an idea what is happening in the brain, meaning initially you would be expecting some random decoding accuracies at negative times because our brain hasn't seen any stimuli yet, so there shouldn't be any information. But then over time, you often see decoding accuracies that are quite high, meaning that there is information in our brain signals showing how the brain encodes these different stimuli. And there is one more way to see this decoding procedure as a decoding matrix. I'm spending a lot of time in this slide because I think it is very important to understand everything that follows. So in this case, we presented only two stimuli, but in general, we conduct experiments by presenting several different stimuli. So we can repeat this procedure pairwise for every combination of two stimuli and populate this decoding matrix that will be indexed by stimuli. So we present 100 stimuli, this would be 100 by 100 matrix. Every element here would be how well we can decode the two elements. And of course, this matrix will also be time resolved, meaning that at different times we will have a different such matrix. And let's see now how we use these ideas to study human vision. And that's a very popular area of research, as I said, because it is highly relevant now with deep learning and advances in visual models that are competing human performance. Really want to see how the human brain relates to machine learning and whether there is a fruitful collaboration between the fields. So from the moment light hits the retina, information travels through the lateral geniculate nucleus, a deep structure in the brain, to early visual cortex. And that's where we have rapidly the initialization of a cascade of activity traveling through the temporal cortex as information is transferred from pixel level information to more high level information in the human brain. V2, V4, these are more high level areas, and eventually IT or inferior temporal cortex, posterior, central, and anterior areas that contain areas in the brain that are very, very dedicated to processing specific stimuli. So our visual brain has a very dedicated visual processing system to study the signals that we experience every day. Faces are very specifically processed in different areas in the brain, scenes, objects, and so on. So we wanted to study these signals with these techniques. So we presented 92 different image stimuli, each of them belonging to ordinal and subordinal categories. So you see here, there are animate and inanimate objects, and with further subdivisions of human and non-human, and then bodies and faces, and we also have natural and artificial objects. So these are concepts that we wanted to see how the brain processes them, whether there are signals that are encoding these concepts in a different way in the brain, and what are the dynamics and where in the brain this happens. So we applied these machine learning tools, and we got these decoding matrices that I mentioned earlier. And these decoding matrices are sort of representations of how the brain sees these different stimuli. We also call them RDMs, representational dissimilarity matrices, because we see how the similar are the different stimuli in these representations. And initially, we wouldn't expect to see any patterns because at negative times, we haven't really experienced the stimuli yet. They're presented at time 0, and soon afterwards, the brain should start responding. Not instantaneously, but there is a small time lag, but then the brain should start exploring this, processing the stimuli, and any patterns that we see here would be indicative of something special that is happening in the brain. So I'm gonna play this video here. Initially, you will see patterns, and also here it's multidimensional scaling, meaning we see these distances here in a 2-dimensional plane, so the different dots are different images, and you see already patterns appearing at these different time instances as the information is processed in the brain, and you see how the different types of stimuli in different colors are clustering together. So indeed, there is some information in the brain regarding this concept that we were discussing before. It cannot be random that the faces, bodies, and inanimate objects are structured in some particular way. Very strongly the faces, if you saw some patterns over there, and then eventually the signals dissipate and become random again. So that's a very powerful technique to see the human brain, even with classical machine learning tools. And thus we can study these representations even more. Now we have these tools. So we have a cascade of these matrices over time, and we can divide these in partitions containing within comparisons of animate, for example, and within comparisons of inanimate, and between comparisons of inanimate and inanimate. And if there is information in the brain regarding animacy, then we would expect somehow animate objects to cluster together, inanimate to cluster together, so these distances should be small, and animate and inanimate should be far apart, so these distances should be large. So if we construct a contrast of these between comparisons minus within, and we do this over time separately for every matrix, we should be able to see some interesting things happening. And indeed, we get a time course of decoding comparison values, showing how the animacy signal is emerging in the brain. And this is not random. If you were to randomly permute the images to exchange labels between animate and inanimate and make such a comparison again, you would really not see such signals and again. So indeed, there seems to be something special happening in the brain, and this is the way we can do statistics as well with permuting labels. And eventually we end up with these decoding time courses showing how, for example, in this case, for the animacy category, the decoding time course peaks sharply at 157 milliseconds. So we can really see the dynamics in the human brain, and we can also see multidimensional scaling, meaning the images are placed based on their relative distance from one another, and you see how faces cluster together. Even a monkey face goes close to the human faces, and other categories like inanimate objects cluster together in different areas. So that's a very powerful method, and it's very general, as you see. So we can apply machine learning in different settings now in neuroscience, in more and more different paradigms, and really see how information is encoded in the human brain. For example, you see another work that studies invariance in human vision, meaning how our brain encodes stimuli, but remains invariant to the relative position or the relative size of the object. So it encodes the concept of the object, and it is invariant to the position of the object, for example. In some way, it's like convolutional neural networks achieve such ideas by doing convolutions in space, being invariant in space, and we have the same ideas in the brain. So this is decoding the stimuli as they are, and this is decoding with size invariance, and this is with position invariance. We can also do decoding in different concepts, such as for face perception, it presents several faces, and we can see how we can decode gender information, or age information, or identity, or familiarity. And this really gives us an idea of which stimuli which properties of the faces are processed first, and which concepts are processed later. So it can also work as a constraint for neural models of vision and so on. And it's not only vision, but we can decode other stimuli as well. For example, here, auditory stimuli coming from auditory sources in different spaces. So we can decode both the source and the space. And we can even decode more complex ideas, such as social interactions, meaning you see images where participants are interacting, where subjects are interacting one another versus they are doing different, attending to different tasks one another, and you can actually decode such conditions. So these methods are very rich in what you can do, and these advances are really, machine learning has been here for a long time, of course, but in this data, it has been the last 6 years or so that we have been looking at these ideas in this way. So it is really an open field to make much more advances in the future. And it goes even more. So there are many imaging methods in neuroscience and neuroimaging. I talked about magnetoencephalography, which is on the left. The most popular one is actually functional magnetic resonance imaging to the right. And though MEG gives you excellent temporal resolution, measuring how magnetic signals change millisecond by millisecond, fMRI is very, very slow in time. Practically, there is not much temporal information because it measures hemodynamic brain activity. There is a change in blood flow when the corresponding brain area is activated. And this takes several seconds to evolve, so it is too slow for the rapid brain dynamics. But we do get maps of hemodynamic changes that are very specific in space. So we have one method that is excellent in time. We have another method that's excellent in space, but we actually want both. And it's really complicated to compare the two because we're talking about physiologically very different signals. However, if you use the ideas I mentioned earlier and construct these representational matrices, which in the case of MEG, they will be changing in time, and in the case of fMRI, they will be changing in space because we can construct these profiles here from local patterns surrounding corresponding brain areas. So in this case, inferior temporal cortex will have its own representational matrix, and so will other brain areas. We can actually compare the two with something as simple as Spearman correlation, so rank-based correlation. And this is what we did. So in MEG, in time, we have these matrices. In fMRI, in space, we have these matrices. So we can exhaustively correlate pairwise these matrices. And by doing so, we can construct these correlation maps, which we call them representational similarity maps. In this case, it will be for time 0, but if you start repeating this for every time, you actually end up with these matrices of dynamic activity. So you can see the dynamics here in the brain. So initially, we don't have any signals yet, but soon after the information reaches in the brain, we start seeing visual signals that are emerging from primary visual cortex back here, and they're rapidly moving ahead in more anterior areas where the brain has the dedicated processing system, like I said, for faces, objects, and scenes. So we can really now see dynamics in a very, very detailed way, something that was not possible before, and thanks to machine learning and extracting such representational patterns. In moving ahead, I want to bring to your attention some conceptual issues when one is doing decoding. For example, we should be careful what is being decoded, because being able to decode doesn't mean that this information is used by the brain. For example, in 2006, in a brain competition, the winning team produced an algorithm that was able to classify different video segments depending on whether they are humorous or serious events, or there was some action and so on. But what it turned out to be is that the critical voxels contributing to this decoding were the ventricles. And ventricles are actually areas in the brain, areas in the head filled with cerebrospinal fluid. So there are no neurons there, there is a fluid there. So we can decode from these signals, but it's really not useful brain signals. What it actually happened is that when participants were looking at humorous segments, they were laughing, and this created motion artifacts which one is able to decode. So we should be careful what we decode, because not all signals are used by the brain. Also, for example, when light hits the retina and travels very quickly in primary visual cortex, at this level, information is still sort of at a pixel level format. So if we were to feed this information to a deep neural network, we would be able to classify almost everything just because they're so powerful. But it doesn't mean that the brain at that point is so it has access to this information. So it needs to process the signals even further down the ventral pathway to extract information. So we should not be using classifiers that are doing something very different from the brain and showing that in V1 we can extract any kind of information that one can decode. So we should be careful at what kind of classifiers we use. And this is why nonlinear classifiers are often avoided in this kind of analysis, though we do want to use them for applications that has to do with prediction, such as, for example, brain disorders or brain-computer interfaces or lie detectors. So we want to use the best classifiers. But when it comes to trying to see what information is immediately accessible by the human brain, it is best to use linear classifiers, because in this case, the information is shown as it is explicitly represented by the brain, meaning that a single neuron at this stage, having us input this kind of information and applying a very basic linear classifier, should be able to extract this information directly. So this is why we used linear support vector machine classifiers before for this kind of task that we want to study the human brain processes. We also should be careful about interpreting decoding accuracies, because they depend on so many factors: which classifier you use, what cross-validation scheme you use, and how many data samples you have, and so on. We need to be careful and balance all these variables so that we can say one condition is decoded higher than another condition. We are also should be careful that we are decoding information in the brain and we don't see activation per se. So for example, here, consider the case that you have a brain region that responds strongly to everything other than faces. So clearly everything is represented other than faces, but if you were to use a classifier, you would actually see that it best decodes faces from everything else. But so interpretation is also a critical issue here. And further, we cannot have directional statistics, meaning we cannot say brain activity is stronger in one case than another one. We can say information is stronger in one case than the other one, but not brain activity itself. Something else important is that how do we interpret the decoding weights? So consider, for example, the case that you have two sources. In one case, you have signal plus noise. In the other case, you only have noise. Clearly, the best classifier here would be the difference between the two. So with weights 1 minus 1, you can really get the signal here. But obviously, there is not a signal in both locations. The signal is just here, but the strength, the absolute value of the weights is equal here. So the classifiers extract weights that are optimal to enhancing the signal and removing noise, and we should interpret them as such, and not that wherever you have strong weights, there is signal. And that's what we see here. The weights actually have noisy-like patterns because the classifier is very efficient in extracting the best optimal way the patterns, but this is not interpretable brain activity. There is a very very key seminal work by Haufe a few years back showing how we can convert these weight maps to activation patterns in the case that we have linear models, linear classifiers, and we can see the actual activations in the brain producing these decoding accuracies. So this is something that is very important to keep in mind when we want to interpret the source of this information in the brain. So what are the best practices given all these concerns? To understand brain signals, it is important to link machine learning results, decoding results with human performance, because then we know that the brain is using this information. It is also important to study decoding patterns, not just in one or two conditions, but many, to be able to know that it generalizes, and even better to show that the classifier generalizes to novel stimuli that wasn't seen before by this classifier, because this is what shows that what the patterns that we extract are really general. And as I mentioned before, it's also a good idea to use a linear classifier. Though I think the previous points are even more important to link with behavior, for example, and to use a lot of stimuli. This really points to us that what we extract is information that is really used by the brain, and we get a glance of the cognitive processing in the brain. Okay, so now let me change gears and show how deep learning can also facilitate in the studies that we do in neuroscience. In this case, we can see how it can contribute to human vision. So we trained in this project a deep neural network. It really doesn't matter what type. It's a convolutional neural network with 8 layers in this case. It was trained from the ImageNet dataset, so several object categories and several images per category. And the idea was to see what kind of representations are created by this model and whether they bear some relation with what the human brain is doing. What we did in every case for humans and machines, we have the same inputs, the same stimuli, but we have different measurement space. So for MEG, I already mentioned that we have magnetic signals. For fMRI, we have hemodynamic signals, and for computational models, we have signals that are processed in different layers, for example, convolutional neural networks. So we can create these representational matrices the same way as before, only for the MEG, they will be changing over time. For the computational models, they will be changing in different layers as you move deeper and deeper in layers. And for fMRI, in different brain regions. And now we wanted to see hierarchical correspondence, meaning that as the signals evolve over time, they become more and more high-level information, from pixels to more high-level semantic information. The same with models, from earlier layers to higher layers, there should be some relationship. And for brain regions, from low level to high level, we should also see such correspondence. So we do expect some correspondence between these three modalities, and we can do that by correlating these corresponding matrices, and it should be, by correlating these two, we should be having similarity time courses for each layer with the MEG temporal data, and in fMRI for every model layer to the brain signals in different brain areas. And indeed, we see such hierarchical correspondence. So I will play this movie. As you see, layers go higher and higher, and information is deeper and deeper in the brain, more anterior. So as we move in higher layers, there is a relationship between brain and models in higher level areas. So indeed, the convolutional neural networks have a similar hierarchical structure with brain signals. So the brain vision has a similarity with machine vision, and we see a similar pattern with MEG and models, meaning that we get different time courses of relation between brain and model layers, and higher layers peak at later times in the visual hierarchy. So higher level signals are coming later and later in the brain, and later in layers in the model. So that's one, one brief demonstration of how one can create relationships between humans and machines. And there is a lot of literature here that I don't have the time to cover here. Even more powerful measurements are with monkey recordings. So in this case, we can represent the machine vision with different networks as information moves layer by layer, but also the monkey as well as the human visual system corresponds to different kind of processing stages, starting early in retina ganglion cells and lateral genicular nucleus, deep structure to V1 and more higher levels in the visual hierarchy. And there is a feed-forward processing here, but also recurrent networks, which is important for one to study. So that one can study corresponding relationships between the two, and in particular see how different machine learning algorithms have relationships with the actual neural data collected from invasive measurements from monkeys. So these measurements are more powerful because you get how individual neurons respond rather than collectively on the neurons macroscopically with non-invasive brain measurements. And recent work has shown, for example, that as you improve the performance of different machine learning algorithms in the ImageNet dataset in this case, you're actually getting closer and closer to brain data, meaning that this is brain score, meaning how close are representations of individual neurons recorded by monkeys to neurons from convolutional neural networks or other brain vision models constructed. And this is very important finding, meaning that there is a synergy between optimizing algorithms for visual tasks and getting closer and closer to the brain. This particular work is done by Jim DiCarlo's group at MIT, and it's one of the pioneering groups in the field. Brain score is actually a site where people can create and upload models and compare with existing monkey data and see how close you're getting to the monkey data. So CoreNet-S is the one that has the best performance at this point. And there is a lot more work showing how recurrent connections, for example, are very important to go closer to human or monkey data. Because difficult tasks such as occlusion or different difficult viewing conditions are really need the power of recurrent connections to create strong algorithms. And also there is a lot of commonality, for example, in early representations, Gabor filters and other stuff, which I don't have time to present. But the idea here is that there is a lot of research happening right now reverse engineering the brain and showing how computational models, deep learning models, are getting closer and closer to human data. And the last part in this talk, I want to discuss a little bit about the importance of graphs, brain graphs, and how we can do machine learning on brain graphs. So brain graphs are very, very important in neuroscience, both for the normal brain, because different processes are happening in different brain networks, but also for the pathologic brain, the abnormal brain. And for example, here, I show you Alzheimer's disease, a devastating disease affecting millions of people and really reducing the quality of life. The problems, the biomarkers in this disease start decades before the first clinical symptoms. So even 20 decades ago, you see proteins accumulating in the brain. Amyloid beta is accumulated extracellularly all around the cortex, but also another protein, tau protein, is accumulated intracellularly, mostly in areas that initially are temporal areas, including hippocampus, an area that is important for memory processing. These two proteins, in ways we don't understand, somehow are participating in a process that is compromising the human brain and leading to synaptic dysfunction, meaning that brain function is altered. And it's not altered in a random way. The accumulation of these proteins happens in specific pathways and is accumulated in a very predictable way in different brain networks. So Alzheimer's disease is a brain network disease, and thus we need dedicated algorithms that process signals in graphs rather than regular Cartesian Euclidean spaces, as we often do in deep learning. The hope would be, for example, to have a functional neuroimaging modality that measures signals from the brain, constructs functional brain networks, and these networks would be able to predict the cascade of the disorder and the progression towards the clinical symptom. And this is very important to predict early, because only then we can conduct effective studies stratify populations and really have strategies to intervene and improve the quality of life, even behaviorally, like anything that we can do to improve the quality of life of these patients. And so thus, this brings me to the topic of geographic deep learning, which has to do with the idea of analyzing data that live in non-Euclidean spaces, such as graphs. So the main field of geometric deep learning is graphs, the main type of data. And we have graphs everywhere in life now, networks, including human brain networks that I mentioned before, but also financial networks, protein networks, social networks, and so on and so forth. The study of networks and graphs is really gaining importance as we collect more and more data of these different types. And for geometric deep learning, there are two key approaches that were introduced recently. One has to do with graph neural networks, which are generalizations of the deep neural networks to graph structured data. So in this case, you have, again, different layers processing information. For example, like in convolutional neural networks, you would have different convolutional layers and pooling layers, and a cascade of them as you increase the complexity of the representations, refining the representations for the task at hand. In this case, you have graph filtering layers and graph pooling layers. And graph filtering are basically applying filtering on data that live on graphs. And these are often generalizations of regular Cartesian processes to graph data. For example, graph signal processing has generalized processes such as spectral filtering or convolutions to graph spaces. And for example, graph spectral filtering relies on, one of the methods relies on the idea that convolution in the spatial domain is actually multiplication in the spectral domain. So convolution is hard to do in graphs, but multiplication is easy to do in graphs. So once you move the data in the spectral domain, you can do a multiplication, which is equivalent to convolution. So we have convolutional layers in graphs as well. And graph pooling is effectively making the graph smaller and smaller by applying some clustering algorithms. So these are powerful tools to study brain data, for example, in the form of graphs. There is another approach as well, which is graph embedding. So in this case, you get high dimensional data in graphs, and you're embedding them in a lower dimensional space, producing vector representations. In this case, in a two-dimensional visualization, you can see them like that, different representations. So embedding high-dimensional graphs in low-dimensional graphs, that would be amenable to all sorts of tasks. And this is the approach that we followed here for to create a predictor for Alzheimer's disease. So we wanted to see alterations of MEG brain networks and how it can predict stages of AD. So the data set in this case was 76 mild cognitively impaired patients and 53 age-matched clinical controls. And the MEG data was resting state data collected for 5 minutes. And these data were used to generate brain networks. And these brain networks produce features that would be valuable for classification. But these are complex high-dimensional data, and our aim was to find better representations to see whether different patients and controls can be classified. So we expanded the architecture of GraphGauss deep learning by providing multiple graphs, and then this model included a 3D encoder that which had a few fully connected layers, yielding eventually representations of the mean and variance of brain nodes. Or in other words, we produced embeddings where every node in the network is represented by a multidimensional Gaussian distribution with a mean and variance output by the embedding model. And these representations are much simpler now to study because they would live in a lower dimensional space, L-dimensional space, and the dimension L is something that one can find by using, for example, a link prediction task, seeing, in other words, what value of L will allow us to best predict edges in the embedded space versus the original space. And the best dimension in this case was 16. So it seems that a 16-dimensional network would be optimal for this kind of task. And finally, we use these embeddings as input data for classical classifiers to distinguish normal controls from stable MCI from progressive MCI. Stable MCI were the patients that remain stable after 3 years of the recordings. Progressing were the ones that after 3 years, they moved to AD, Alzheimer's disease. So we were able to distinguish all this, to dissociate all this in a 3-way classification using, for example, random forest reaching 8-to-1 decoding accuracy. So this shows the promise of using deep learning and machine learning to extract brain signals from graphs. And you can also use this kind of signals to do unsupervised classification, possibly leading to clustering of patients that may reveal subtypes of Alzheimer's disease and mild cognitive impairment, which could be useful clinically if indeed they are accompanied by different symptoms. So I want to stop here. I hope I didn't go very fast in the presentation, and I'm not sure if there are any questions or anything. I'm happy to address them at this point.
Well, I see there is a question on the visual path may be related to visual attention.
Yes. I'm not, should I move the slides, or maybe you can read the question? I'm not sure where to go.
So there is here another question. Apart from images, other sensory stimuli was also tested, like smell, hearing. Can you comment on that?
So my comment is that the tools that I presented as machine learning tools are extremely general, meaning that you get these signals from the brain and you can really use them to decode any concept you want. So, so you can decode image categories, or you can decode even smell or memory or attention or all sorts of different cognitive processes. And indeed, researchers are doing that now. They are really applying these models to study different cognitive processes. I'm not sure. I hope I'm answering this, or maybe if I understood the question well, it's the answer is simple. These are really general tools. I haven't applied myself to all different domains, but I know works from different investigators studying, for example, memory. And this is really amazing, like a working memory. You see how the brain is store multiple types of information for a short time and reusing it.
There is another question. Can you elaborate more on the brain's core? What features of an artificial neural network are exactly compared to natural ones?
So that's a good question. To my understanding, because I'm not, I'm not part of this work, but I am, to my understanding, the deep neural networks also have neurons, individual neurons. And so every layer is basically producing signals for every input you use to this network. So for example, you present the same image to the monkey and to this neural network. You will see how strong a neuron responds to this image, and you will also see how strong a monkey neuron responds to this image. Now, you don't send one image, but let's say you present like 100 images. So the same neuron in the deep learning model will have 100 responses, and the same neuron in the monkey will have 100 responses. So you can really relate the two and see if they have the same patterns or not. So it is the output of different layers, different neurons of different layers.
Okay, great. I see there is another question. So, as far as I understand, sensors on MEG record a collective neuron activity. Have you tried to apply source separation techniques to obtain better resolution?
Yes, that is a good question. And the strength of MEG, one strength of MEG is that it gives you better spatial resolution than the sister method, which is EEG. So yes, we oftentimes apply these tools in brain regions as well, rather than on the sensor space. My preferred approach is usually to study the dynamics. So these algorithms work well despite the high dimensionality of the data, 300 variables. So I oftentimes get the time courses, the decoding time courses, and then focus on pretty interesting time points to see the origin of these signals. But one can definitely apply these tools on the cortex as well, or even differently, decode in the sensor domain, but then use the mapping tools that I described before, because there is a linear mapping from the sources to the sensor and a linear mapping from the linear classifier itself. So linear mappings allow us to convert the weights of the classifier to activation patterns. So in advanced, ways of analysis, one can even decode in sensors and see the corresponding activation pattern on the sources. So there are different ways to go, but yes, the ultimate aim would be to learn not only temporal information, but also spatial information.
So I see it seems there are no more questions. I don't know if someone wants still to ask something. Now is the right moment. Well, it seems it is everything, and also we are finishing nicely on time. So
Okay, this sounds all great. I mean, it was a pleasure, like I said, to be part of this effort.
Yes.
And I want to congratulate you guys for organizing this. And thanks everybody for attending.
Thank you very much, Demetrios. And if someone has questions, wants to contact you, what is the easiest way to reach you? Email?
Yes, definitely. Please email me. I will be happy to respond.
Great. Well, then, thank you very much, and we wish you a nice day.
Thank you, you too.
Bye.